would be helpful. Hi, Ian. Oh, perfect. All right. Okay, so plan for today. We are going to do, I shouldn't say we, you guys are going to do some notes today. We're going to go over the slide 6 from yesterday, the one that I set you guys off to do independently. We'll go through that. We'll do like corrections and whatnot, and then I'll get you started on your notes. So you'll have independent time to work on that, and then tomorrow we'll go through the notes and do a whole recap. You'll have like an end of the week, I don't even call it a quiz, like big exit ticket kind of a thing, just recapping like what you would need to know. Make sure you guys are all on the same page for after the end of the week, especially with the nucleic acid stuff. Monday being our shorty period again. Want to make sure that before we transition over to the next thing that everybody is squared away. So to get us going, in the chat, please answer the following question. So what are the 4 types of nucleotides or building blocks that make up DNA? Hint, it's the letters. So what were those 4 letters that you guys used yesterday to build out your DNA models? Hi, Hayla. So Gavin thinks it's A's, T, C, and G. Hi, Lynn. Basically, if you guys all just copied what Gavin and Rylan said right now, you'd be spot on. Good job. Please remember to participate a bit. It makes the class and the world go better. Now, yesterday, when you guys watched the video, they did talk about what those, what the proper names of each of the building blocks were called. So A stood for adenine, T stood for thymine, C stands for cytosine, and G stands for guanine. I will tell you on your test, I will give you both. So I will have the word, the actual name there, but for all intents and purposes for the rest of the unit, we're really just going to be referring to them as A's, T's, C's, and G's. But do remember they actually have a name. But for today, for your goal, we're going to be building off what our goal was yesterday. So if you remember, our goal yesterday was, can you define DNA? And can you define the word nucleotide? Okay, so remember, DNA was our source of our genetic material. Nucleotides are the building blocks of DNA. Today, you're going to be adding RNA to your definition list, okay, or your vocab list. Yesterday, you modeled the structure of DNA, which we're going to talk about here in a second. Today, you're going to add in RNA. Then you guys will use those models to explain, especially that relationship between structure and function for the DNA. And you guys are going to compare and contrast both, so DNA and RNA. So how are they similar and how are they different? Okay, remember, when we get into talking about the nucleic acids is a family of molecules. So we're going to be talking about it from that perspective here tomorrow in terms of almost like a fun mafia family where DNA is the boss. It is the godfather of the nucleic acids. And just like all proper gangster families, they have a focus. Like, how do they make their money? What is their job? What is their big thing? In terms of our nucleic acids, it's proteins. Okay, proteins is their business. So we're going to kind of do everything with that focus. So how do they run their business? How do they get things done? What does that organization structure look like for them? Okay. Before we get going on our notes, though, let's go back to slide 6. Okay. And I'm going to present this so it's a little bit bigger for you guys. So you guys should have, again, this is pretty much giving you guys the answers, but you should have watched this. It will be of no help to you if you didn't and you're just copying stuff down. You still won't get the answers right on the test. But we're going to go through each one of these and kind of go a little bit more in depth with it. So like we talked about yesterday, when we look at that first question in the table, and first question number 3, DNA contains the code for making what? We kept going back to this idea, and I kept repeating, like, it's instructions for you. It's the code for making you, everything about you. Well, really more specifically what we're looking at, it's the code for making all the proteins. And you are the product of your proteins. There is not a single thing that happens in your body that does not rely or have something to do with a protein. Okay, so whether that is building muscle, repairing things around your body, managing chemical reactions. So you are one walking chemical reaction, and it's because of proteins that's able to happen. Sending messages throughout your body. That's the work of proteins. So it's no coincidence that up until the 1950s, we all thought it was proteins that was the source of your genetic information. No, but they weren't that far off. Okay, so while we know now DNA is your genetic code, your instruction manual, really that's where the link is. It's the instructions for making all your necessary proteins, because without them, you're a donezo. There's just not a chance. Okay, you are one giant pile of proteins. Okay, now when we get into this next piece where we start to look at nucleotides as being their building block of this molecule. Yesterday when we made like the puzzle, we were using blocks or puzzle pieces as one solid unit. That was just for simplicity's sake, because the nucleotide itself is a 3-piece unit. Okay, so it's made up of 3 smaller pieces to form one big one. And the 3 parts are going to be a phosphate group, which is the circle right here that we're going to use, a sugar, specifically deoxyribose. Now, whenever you see anything like this, where you see a pentagon or you're going to see a hexagon, some sort of what we call a ring structure, right here, like a geometric shape like this, that's going to represent sugars. Okay. And then ultimately you have this, what we call a nitrogen base. You'll also, it's okay to call them just bases. So what are your bases? Now, the way that these nucleotides are connected really is that a good representation of that whole structure function argument here. Now, these guys, this side right here, when you have a phosphate sugar, phosphate sugar, phosphate sugar, that is what we refer to as the backbone of DNA. Now, think about your backbone. What does your backbone do for you? So what do you guys think? What are some things your backbone actually does for you? Yeah, it allows you to move. It allows for movement. It also gives you some like form, right? Some stability, some structure. Think about if we removed your backbone, you would essentially be like a pool of jelly, like a jellyfish. Okay, we would be invertebrates. We would have no spine, so we would have no form. Okay, the backbone of the DNA it not only provides stability and support, but it allows that DNA to be manipulated and to be shifted and moved around to change shapes, and it also provides protection to your code. Okay, each of the strands of nucleotides, because if you kind of take a look over here, I'm kind of doing these two right now. Yes, DNA, that's true, is double-stranded. So you're going to have one strand of nucleotides that link together and another strand. Each one of those strands has a backbone. Okay, so think very much like a book. So if you had a textbook, you have one hardcover on one end and another hardcover on the other end, and it protects the pages in the middle. That's the info. That's the good stuff. That's what your DNA is doing. It's protecting all of this good stuff here in the middle. This is the instructions. We need to keep this nice and safe. Now, some of the ways that your DNA does this is by remember it spins, it's a double helix. So double meaning 2 strands that are going to literally form the spiraling effect around each other. Now, the reason why it can do that And one of the other things it can do, so think like a slinky, you can compress a slinky down and you can stretch it out, and then it'll squish back down and you can stretch it out. That is because of these guys right here and how they're linked together. Your phosphate groups, think of it like a ball joint. Okay, so it's going to be some sort of joint that's going to allow, if this was the joint right here where my wrists are, it's going to allow that molecule to open up, so expand, collapse down, as well as turn to create that helix pattern or that helix shape. Okay, so the anchor points for the backbone kind of are right here with these, what we call the phosphate groups. And a phosphate group is just a collection of phosphorus atoms, oxygen atoms, and hydrogen atoms that kind of create a little cluster and a unit. And they're kind of linking and holding on and kind of allowing this like pivoting motion for the DNA. Now your sugars are what link to that little ball joint, that little pivoting structure. And those sugars, their primary job, not only to be a part of the backbone, but notice that they hold on to the nitrogen base. They're holding on to the code. So this is the link between the code and the actual backbone. So that right there, if that sugar wasn't there, there would be no way for the backbone to be able to hold on to that nitrogen base there in the center. Okay. So again, this kind of allows it to spin, to condense, to spread, and kind of it works like this little motion. So it can go boop, 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 boop. Okay. Now, wherever you see these straight lines, okay, all up in here, those represent what we call covalent bonds. And covalent bonds are, of the bonds, they're one of the stronger ones. And they occur when you have two atoms that are going to share some electrons. And by sharing, it keeps them really close and connected. And that's really important to the stability of the backbone. You want to have strong bonds holding to your backbone together, right? If this was made up of weak bonds, you just crumble and fall apart and it would be a waste. Okay, so while you have these stronger bonds on the outsides and holding on to the actual code, right here in the middle, DNA uses a different type of bond. And this bond, we don't want to be very strong because you need to be able to access that code. So it would be like you use that book analogy, okay? You have those strong bonds holding together the outside covers, okay, and the spine, and you open up, this would be like the pages being all sorts of glued together. There's no way you're going to be able to read that. So instead, what these bonds do is allow this like really flexy, like you're closed, you're open, you're together, you're not, you're together, you're not. Hydrogen bonds are weak bonds and they come together and break apart really easily. It does not take a whole lot of effort to make that happen. So you can think of it also kind of like a zipper. So like your zipper on your jacket, you can go up and then you can go down. You can go up and you can go down repeatedly over and over and over. That is exactly what these hydrogen bonds do. When they're together, they seal up the code and allow the DNA to like trap it in there. But when that code needs to be read to make some proteins and that DNA unwinds itself and opens up, those little weak bonds that are holding these two strands together separate so that you have those different proteins come in and they can read the code. And then when they're done reading, they close back up, they reform, and then that DNA winds back down and squishes back together. Okay. Now, the big difference here when you're looking at the connection points is that A's and T's tend to be just a little bit smaller. So they're only going to need to use two hydrogen bonds to hold them together at that spot. While the G's and C's, your guanine and cytosine, they're just a little bit bigger on the sides. They're going to need 3 bonds to hold them together. Okay. And this last one we chatted about yesterday, how are we going to remember that A's pair with T's and C's pair with G's? Remember, they even talked about it in the video. When you have an A, you use straight angular lines. So does the T's. You're using straight lines to make those. So A's and T's go together. And then when you make a G and a C, you're using curvy lines. Okay, so that's kind of an easy way we can remember that. G's and C's always go together for DNA. A's and T's always go together for DNA. Okay. Now, what you guys have here, all those labeled pieces, this is showing you where you have two nucleotides stacked on one strand. Remember when we talk about the size of this thing, one single strand of DNA can be about 3,600 nucleotides long. That's a whole lot of code. Okay, and that's a whole lot of stacking here that we have to condense. All right. Questions on slide 6, or the structural pieces for DNA. Okay. So we're now going to go back and we're going to skip over some slides and we're going to jump to slide 13. Okay, so in Google Classroom, you've been assigned a Nearpod where you're going to use the information in the Nearpod to answer the questions or answer the two sides on slide 13. So the right-hand side and the left-hand side. On the right-hand side, you're going to talk about the important facts about DNA. So things like what's its function, where is it found, and then other important facts. Remember, the nucleic acids specifically, that family, are all about making proteins. And each one of these members, so DNA and all the different types of RNA, have a role in that process. Try to steer your notes to focus on their role in the process of making proteins. So how does it get involved in that process? What's its role? Okay. I have added these guys over here again for you. These are totally optional. It's just if you run out of space in this side, you can kind of tuck these on over. Remember, these are like Post-it notes. If you end up not using them and you don't like them there, they bug you, you can go ahead and delete them. That's totally fine. But the second thing you're going to look at is the function of your RNAs. Now, there's 3 specific types of RNA that you guys are going to be looking at. And they are what we call mRNA, rRNA, and tRNA. And each one is essentially like a cousin of the DNA. Okay, they're all related. Now, this little lowercase letter at the front of each one of those has something to do with its job. Okay, and that's how we kind of we categorize them. So we kind of label them in that fashion. Okay, so what is the role of the mRNA? So what's important about it? What does it do for the production of proteins? What does it do for the family of nucleic acids? Same thing for your RNA and same thing for the tRNA. Okay. After you've got all that, then you're going to go to this left-hand side and you're going to fill out a quick compare and contrast for the DNA and RNA. Try to get at least 3 different things for each column. So 3 things that are unique for DNA, 3 things that are only for RNA, and then 3 things that they all have in common. Okay, so remember, just like a family, you're different from your family in a lot of ways, but you're also very similar to your family in a lot of ways. So you can add more than that. Tomorrow during our discussion, I will put more than 3, but try to find at least 3 for each column. Okay. All right, last piece. When you go to your, I'll just use 5th period, that's fine. When you go to the post for today's lesson 2, so explain part 1, nucleic acids, you're gonna see the link for the Nearpod. Now, I know some people's computers kind of trip out using Nearpods, so I removed the requirement that you can't skip ahead. So you can skip ahead. So if your Nearpod always gets stuck on the videos, it just sucks at loading them through Nearpod, totally get it. What I did is I added those videos that are embedded in the Nearpod to the post. So if your video gets stuck, don't sit and struggle with it, move past it, and then come back to Google Classroom and then watch it through here. Okay, we'll click on one of the links for the videos here. The only other thing that was embedded within the Nearpod is a short little reading that you're going to use that discusses the RNAs and the DNAs, okay? So again, these are not extra like new videos. These are the same ones in here. It's just if the Nearpod, if it struggles for you, okay, you guys can jump on out of there and watch them through here, okay? So once you've got that all taken care of, you are basically done for the day. Tomorrow we'll discuss slide 13 altogether and get all of those important facts about your nucleic acids kind of down. So your job today is to complete complete slide 13 in your notebook, and then also make sure that slides 5 and 6 are complete as well. And that would make sure that you are up to date and you have completed all the sections that you need to this far, okay? If you guys have questions, you can stick around and I can walk you through anything you guys need to. But if you feel good about what your job is for today, remember, I know you guys have practice and whatnot for some of you. This just needs to be done by tomorrow when you come into class, okay? So if you're good to go, you are free to go. You do not have to stay. Make sure you're getting your work done, and I'm here to answer all questions until 2:45 for you, okay? Bye. Bye, Amanda. Bye, Lynn. Bye, Ellen. Bye, Isabella. Bye, Roman.